right, class. So we can start now. It is 10 o'clock. These are some very good references on CRISPR that you might want to go back and refer to. So this is 2020, and this is Nobel Prize for Emmanuelle Charpentier and Jennifer Doudna. This Nobel Prize has been given for discovering and transforming CRISPR into a gene editing technology. And this is the first time two females have shared a Nobel Prize for any subject. And we talked of this earlier also. What is the genome? Genome is one full complement of genetic material in an organism. And editing genome means you're trying to change the sequence of nucleotides, which has been naturally inherited by the individual. So basically, you're trying to alter the sequence of nucleotides, ATGC, in a piece of DNA, which has been naturally inherited. And this changes occur also naturally. It occurs naturally via errors during DNA replication. And I've told you DNA polymerase has high fidelity, and it also has a proofreading function to ensure that if a wrong nucleotide has got incorporated, it can be corrected and the correct nucleotide can be incorporated. But at times, even after this strong fidelity and proofreading function, you have incorporation of wrong nucleotides, and that is responsible for what is your single nucleotide polymorphisms that may arise because of errors in DNA replication also. And then, of course, they can also arise from because of spontaneous mutations. Then you can have errors during crossing over. So there is, crossing over happens and there is reciprocal exchange of segments that happens between homologous chromosomes. But at times what happens is that this exchange is not reciprocal. So that is errors during crossing over where large scale genome editing can happen. Then, of course, the spontaneous mutations. Mutations have a tendency to appear at a certain frequency in the population. So these spontaneous mutations can also cause a single nucleotide edits. Most commonly, it could be a insertion, it could be a single nucleotide deletion, it could be a substitution. So those kind of things can happen here. And then, of course, DNA damage and repair. When the DNA is damaged, there are two ways of repair, as I told you earlier. One is the homology-mediated repair, where you refer to the true copy, the other copy, and then repair the DNA. And the other type of repair is where you have non-homologous end joining. So you have two ends that have been broken. They are simply joined by removing whatever extra nucleotides are there or inserting a few extra nucleotides. So that is error prone, and that can result in a lot of genome edits. And then, of course, why do we want to edit genomes? We want to edit genomes for several reasons. One, of course, is we want to correct a genetic inborn disease by going into the DNA and specifically correcting the nucleotide, that the mutation that is responsible for that particular disease. While we are talking of single nucleotide mutations, but also you can replace the entire wrong gene copy or the faulty gene copy by the correct gene copies. Then, of course, you want to investigate gene function. So one of the major ways of investigating gene function has been to basically generate a loss of function mutation, where the protein now or the gene and the protein are non-functional because the functional protein is not coded. And then you compare for whether there is, what is the difference between normal and the mutant mouse. And or for that matter, other models can be used. For example, you can use zebrafish, used most commonly for vascular development, C. elegans flatworm model, which is used most commonly for aging experiments. And then, of course, mouse and Drosophila have been our standard model organisms. Then, of course, you want to investigate the protein function and protein localization, its interactive partners, and so on and so forth. So you can add a fluorescent tag to that protein of interest. Now your protein will fluoresce. So wherever on the cell it goes, it will be able to give you a fluorescent signal that allows you to identify the movement of the protein, and also it can help you identify which other proteins it is co-localizing with, and probably because it is co-localizing, it is interacting with such proteins. And then, of course, in certain cases, you want to enhance the gene function. You want to basically express more of the protein. You can use genome editing to try and enhance the gene function, and we'll see how that is done here. Right? So what is CRISPR-Cas9? Basically, CRISPR-Cas system. CRISPR-Cas system is an adaptive immune response in bacteria, causing double-strand breaks in the DNA of invading bacteriophage or plasmid, mediated by non-coding RNAs, CRISPR RNA and TRISR RNA, and Cas endonucleases. So this is basically an adaptive immune response. Why is it adaptive? Because it creates a memory of the previous attack, and if the same bacteriophage is attacking again, then the response is much quicker and faster. If a new bacteriophage is attacking, then of course the system is slightly different. Instead of Cas9, Cas1 and 2 take part in that kind of interaction. And we'll see how that is done. There are several types of CRISPRs, but most common is type 2 CRISPR, is one of the most well-characterized systems and carries out targeted DNA double-strand break in 4 sequential steps. Principles are simple. One is the base complementarity rule. This is a very simple and elegant method whereby two nucleic acids that are complementary to each other, and the two will find each other and anneal with each other. So this is one example where the two of equal length. And then you have one example more here. This is where you have a longer one strand and you have a very short, the second strand is very short, but still they are complementary to each other and therefore they will come and bind to each other. So this is one very elegant natural design to produce a copy, right? So this is the, I mean, you have to appreciate that over the billion of years, life has evolved and it has basically evolved the best strategies to produce a copy of the original material. And this is the base complementarity rule, right? And then, of course, we have endonucleases. Endonucleases are enzymes that cleave the phosphodiester bond with a polynucleotide within a polynucleotide chain. So endo means within, and nucleases means they are going to digest nucleic acids. And one of the common endonucleases that you have read about is restriction enzymes of class 2. A restriction enzyme class 2 basically cut within a specific recognition sequence. There has to be a specific sequence in the DNA, and if that is present, the restriction enzymes can come and cut there, and then they can produce a staggered end or a blunt end, depending on how they're, what is the nature of the enzyme. In this case, in case of CRISPR, the endonuclease that is used is called Cas9. This is guided by RNA, and wherever the RNA sequence is complementary to the DNA, that is the position where this Cas9 is going to cut. So these are the basic components. Now we move on, and we also talk of a bit more of DNA repair mechanisms. There are two mechanisms. One is your homologous, the homology-directed repair, where whenever a strand break happens, this piece of DNA can refer to the other homolog and see what is the sequence there, and then copy the correct sequence from here. This results in repair of the DNA break. And then the other is non-homologous end joining, where there is no reference sequence there. And what you have directly is there is a double strand break. Whenever there is a double strand break, there will be the sequence will be partly either deleted or new nucleotides may be added, and then they're simply ligated to each other without any reference. So this is a lot more error prone as compared to the homology-directed repair. And if you look at the homology-directed repair, you can force a homology-directed repair on cells by adding a homologous piece of DNA. So here is the homologous piece of DNA. What you want is basically this is the endogenous DNA, the cell's own DNA. This DNA has a non-functional gene copy. And what you want to do is to introduce a functional copy into this non-functional. So you want to replace the non-functional copy by the functional copy. So what you do is you introduce a construct. This is basically a DNA sequence that contains your correct copy of the gene, and it has flanking regions which are exactly similar sequence to the region flanking the copy that you want to replace here. If you see here, this is brown coded, this is brown coded. These are exactly similar sequences. So when this piece of DNA is introduced, this is going to go into nuclease, and then because there is recombination that is going to happen here, this correct copy gets inserted, and the faulty copy is basically excluded out, and this is unstable and is degraded. So now what you have done is effectively using homologous recombination, you have specifically removed a part of the DNA and replaced it by the copy that you wanted, right? And this copy could be your correct copy or could be faulty copy, depending on what is your experimental paradigm. If you want to check for gene function, then of course you want to basically generate a knockout. So knockout means you will have to basically replace the functional copy by a non-functional copy. So here is your CRISPR locus, and if you look at the CRISPR locus, basically it consists of one, the CRISPR repeat spacer array. So this is one part of the CRISPR locus. Then upstream of this, there is the CAS locus, which consists of the CRISPR associated genes or CRISPR associated proteins. And upstream of this, there is a transacting RNA locus or tracer RNA locus. And if you look at the CRISPR repeat spacer locus, this is where the name comes from. Cluster of regularly interspaced short palindromic repeats, right? So this is a cluster here of several sequences. What are the sequences that you have here? You have a repeat sequence that is regularly interspaced by a spacer sequence. These spacer sequences are unique sequences. Repeat sequences are exactly the same sequences which are coming at a regular interval, and they are broken in between by what is known as the spacer sequence. And this spacer sequence basically is a signature sequence coming from a previous infection of bacteriophage. This repeat sequence is a palindrome sequence, and it is interspaced by spacer sequences. Spacer sequences are unique sequences, repeat sequences similar across. And therefore, if you see, this is represented by turquoise squares here of the same color, and then the spacer sequences are represented by different colors. And how are these spacers coming from? These spacer sequences are coming from a part of the DNA that has been plucked out from the previous bacteriophage infections. And we'll see this how this is done. So, and then of course you have the Cas operon, which is basically the operon is the type of gene organization found in bacteria where genes of the same pathway and genes of the same pathway are functionally and spatially clustered together. So they are basically all put together and they are under the control of single promoter. So either all of them are expressed together or none of them are expressed. So this is basically the advantage with operons. And here you have the Cas operon or cluster associated genes operon. And you have Cas9, which is the most common endonuclease that is used here. There are other Cases also that are used. And then of course you have Cas1, Cas2, which are used to acquire these spacers. There are two things that you're doing here. One is acquiring the spacer if there is a novel infection. And the second is also checking for whether the infection has already occurred. And therefore you already have a signature sequence in the spacer here, right? And then, of course, you have the tracer RNA locus, which is the transacting RNA, facilitates conversion of pre-CRISPR RNA to CRISPR RNA and formation of CRISPR-Cas complex. Approximately 45% of all bacteria will have the CRISPR complex, and almost 85% of all archaea have the CRISPR complex, right? So this is an adaptive immune response system. How does it work? So first, if you see, as and when a virus attacks, first thing is there is a transcription of the CRISPR repeat spacer array, forming a pre-CRISPR RNA, right? So this is the pre-CRISPR RNA that is formed. Pre-CRISPR RNA is then broken into a spacer and repeat. This is one CRISPR RNA, this is the second CRISPR RNA, this is the third CRISPR RNA of different spacers. So you see the spacers are different here, and the repeat region is still the same, represented by the same color. And to the repeat region, the tracer RNA can bind here with partial homology, so therefore with partial matching, and therefore the tracer RNA comes and binds to this repeat region, forming the CRISPR tracer double di-hybrid, di-RNA hybrid or double RNA hybrid. So if you see here, and then finally they recruit Cas9 together. So finally what you have is a ribonucleoprotein consisting of two RNAs and one protein. The protein in question is Cas9. The RNA here are CRISPR RNA. CRISPR RNA has the spacer region and the repeat region. And the other RNA is the tracer RNA, which is here. Tracer RNA is partially matching with the repeat in the CRISPR RNA, and therefore they can hybridize at this position. So this forms a partially double-stranded structure here. The process that happens is like this. So as I said, two non-coding RNA, the pre-CRISPR RNA array and tracer RNA are transcribed from the CRISPR locus. The tracer RNA hybridizes to the repeat region of the pre-CRISPR RNA and mediates the processing of pre-CRISPR RNA into mature CRISPR RNA. So basically this process where you have the breaking of this pre-CRISPR RNA into individual CRISPR RNAs is mediated by tracer RNA. So tracer RNA comes and binds here, here, and here in the repeat region. And then it also facilitates the breaking of these long CRISPR transcript into smaller CRISPR transcripts, which are the final transcripts here. And then, of course, you have the other enzymes that play a role. There are several unknown nucleases that play a role and then play a role in assembly. And finally, what you have is a ribonucleoprotein, which is consisting of two RNAs and one endonuclease. Endonuclease is Cas9, and the two RNAs are CRISPR RNA and the tracer RNA here, right? And then this, because now this ribonucleoprotein has a spacer sequence which is unhybridized, which is single-stranded, this now will scan your foreign DNA, and wherever it finds a complete match, ideally it should be a complete match. One odd mismatch may be allowed, so there can be off-target effects also for CRISPR. But generally, what is found is this is much more specific than previous technologies that were there. So now this will scan the invading DNA, and wherever the spacer region finds a match on the target DNA, that region is known as protospacer on the target DNA. There, Cas9 will introduce a double-strand break, resulting in the loss of certain nucleotides in the DNA, and therefore now the virus attack is basically cancelled or annulled or basically thrown back, right? So it is repulsed. So the virus cannot then now finally integrate into the bacteria and multiply and whatever. It cannot overtake the bacterial machinery for invasion and for multiplication. So this is the whole process here. Let me just take you one more diagram that I've added so that it is very clear here. This is your CRISPR RNA in blue. The dark blue part is the repeat region and the light blue part is the spacer region, right? And this dark blue part is partially similar to what you have in tracer RNA. So these two hybridize, forming the tracer RNA, CRISPR RNA, dihybrid RNA that is there, which is partially double-stranded, and then of course it is single-stranded. And then this then attaches with Cas9, and then this scans the target DNA for complementarity to the spacer region here. So this is your spacer region. It is looking for complementarity in the target DNA. Wherever it finds a complementarity in the target DNA, and most commonly what we want is absolute complementarity, means if this is around 20 nucleotides in length, the spacer region, all 20 nucleotides should be exactly complementary to the nucleotides on the target DNA. That region is known as the protospacer region, and this is where the Cas9 is then going to induce cuts on the two strands of the target DNA. And opposite to the protospacer region, which is part of the target DNA, you have the other complementary strand to this, and here you have what is known as the PAM region, or protospacer adjacent motif. So the part of target DNA that is complementary to the spacer is called protospacer. On the opposite strand of the target DNA, you have a motif that is known as the protospacer adjacent motif. So as the name indicates, protospacer adjacent motif, it will be next to the protospacer on the opposite strand, right? And this is basically species specific. Each species has a specific protospacer adjacent motif. In case of Streptococcus pyogenes, where CRISPR-Cas9 has been developed, this motif is NGG, or NGG meaning N means any nucleotide, and then followed by a G and a G. So this is basically your protospacer adjacent motif. And this PAM motif is essential for Cas9 to induce a double strand break. If the PAM motif is not present, you will not have a break in the DNA, right? So therefore, you have to make sure when you're selecting your region for where you want to target your DNA, it must also have an adjacent PAM motif. That is one additional requirement. So in the natural system, if you see, CRISPR-Cas9 complex consists of 2 RNAs and 1 protein. Right, class? Class, you follow? In the natural systems, CRISPR-Cas complex consists of 2 RNA and 1 protein, right? So this is something that you need to keep in mind. And then we move on and we see here again one more time, and this time we talk in more details of Cas9. So you have the CRISPR RNA. CRISPR RNA consists of a repeat region and a spacer region. You have the tracer RNA. Tracer RNA is partially similar or partially complementary to the CRISPR RNA, and therefore it hybridizes, forming a dihybrid RNA complex here. And this then, with Cas9, this complex then scans the target DNA for regions of complementarity. Wherever there is complete complementarity, Cas9 has 2 domains that can cut. One is known as the RUVC domain, and the other is known as HNH domain. RUVC domain is going to cut on the strand that has the PAM motif. So this is a PAM motif here. So it is basically cutting on the opposite strand. And the HNH domain is going to cut on the protospacer strand here. So this is your protospacer here, which is complementary to the spacer. So the region of the target DNA that is complementary to the spacer region is the protospacer. So here it cuts, the HNH domain cuts on the protospacer, and RUVC domain cuts on the other strand, giving rise to a double-stranded break in the DNA, resulting in the nullifying of the viral attack. The mature CRISPR tracer RNA complex directs Cas9 to the target viral DNA via Watson-Crick base pairing between the spacer on the CRISPR RNA and the protospacer on the target DNA next to the protospacer adjacent motif. So this PAM motif has to be present, right? This is an essential requirement for Cas9 to work. Apart from the complete complementarity between the spacer and the protospacer, you must also have a PAM motif or protospacer adjacent motif, which is specific for each species. And that again is important. So that has to be present. So therefore, not every region in the DNA can be directly targeted using CRISPR. It has to have a PAM motif. And what we now do is to, make this PAM motif slightly engineered so that a lot more flexibility can be there. So there are 2 nuclease domains in Cas9, HNH nuclease domain that cleaves the target DNA or a target strand of DNA, and RUVC, which cleaves the non-target strand of DNA, right? In order for Cas9 to function, it also requires a specific protospacer adjacent motif that varies depending on the bacterial species of the Cas9 that you're investigating. What is CRISPR-Cas9 basically? CRISPR-Cas system. CRISPR-Cas system is an adaptive immune response in bacteria, causing double-strand breaks in the DNA of invading bacteriophage or plasmid, mediated by non-coding RNAs, CRISPR RNA and tracer RNA, and Cas endonucleases. So this is basically an adaptive immune response. Why is it adaptive? Because it creates a memory of the previous attack, and if the same bacteriophage is attacking again, then the response is much quicker and faster. If a new bacteriophage is attacking, then of course the system is slightly different. Instead of Cas9, Cas1 and 2 take part in that kind of interaction. And we'll see how that is done. There are several types of CRISPRs, but most common is type 2 CRISPR, is one of the most well-characterized systems and carries out targeted DNA double-strand break in 4 sequential steps. Now coming on to, let's say you have a CRISPR locus here. So this is basically a signature of all previous bacteriophage infections. If a previously known bacteriophage is infecting again, then CRISPR-Cas9 will go and digest the DNA. But if there is a new virus that is attacked, then in that case, the viral DNA is released into the cytoplasm of the bacteria, and the bacteria in response would then release Cas1 and Cas2. Cas1 and Cas2, in contrast to Cas9, which basically cuts at a specific region where the spacer is complementary to the target DNA. In this case here, this is sequence non-specific. So Cas1 and Cas2 can cut any DNA without any specificity for a specific nucleotide sequence. So they can cut anywhere within the DNA. These are endonucleases again, and they can cut anywhere within the DNA. So therefore, they would go and cut. And what they also do is they're not just endonucleases, they're also integrases. So in the sense that after they've cut a piece of DNA, they would also integrate that piece of DNA into this CRISPR array here. So a new novel spacer or a new sequence coming from a new bacteriophage infection would now come and get stuck here at the new place here. So this is the first infection, second infection, third infection. So if you look at the CRISPR array, you can actually deduce the history of infections of bacteriophages in a given bacteria. You see the beauty of the system class, right? This is absolutely amazing. Absolutely wonderful system, a very simple, very elegant, very innovative and intuitive system. Remember, this is all common sense, right? In biotechnology, you can go very far if you have common sense. When I joined my PhD in CCMB, I did not know anything about biotechnology. I was a pure MSc zoology postgraduate from Lucknow University. So I had not even seen a pipette then. But then what kept me afloat was the common sense and the basic idea that there has to be a rationale behind what is happening in an experiment. So the idea is that you have to be a bit more intuitive, because biological systems are based on very intuitive, very elegant designs. Because remember, these are systems that have been perfected over millions of years of evolution. So these are the best systems that are known. So coming back here, class. This is what happens in case of a novel virus attack. In case of a novel virus attack, you will have your Cas1, Cas2 complex that will go there and digest the viral DNA, generate a signature of that, and that signature will be added to the CRISPR array here, right? So the last signature that you get would be the latest infection that you've got here, right? And if it is already a known infection, then of course Cas9 and your CRISPR will go and digest that DNA. So coming back here, we talk of protospacer adjacent motifs. So this is your, if you see here again in this diagram, this is your spacer. This is your repeat region, which is complementary to the transacting RNA and therefore tracer RNA, and therefore this is your tracer CRISPR hybrid. In the CRISPR RNA, this is your spacer region. This is complementary to a part of the target DNA. This is your protospacer. And the strand opposite to the protospacer is where you'll have your PAM region or protospacer adjacent motif. What is protospacer adjacent motif? It is a particular sequence combination that has to be present for Cas9 to activate a double strand break. So this is an absolute requirement. Without this, Cas9 cannot work. So therefore, when you're targeting a region, you have to also ensure that it has a PAM motif nearby. It has to be within 2 to 3 nucleotides or 5 nucleotides of the recognition sequence that is there, of the region where this is complementary. So the target sequence that is complementary to the spacer sequence is known as protospacer. In order for Cas9 to function, it also requires a specific protospacer adjacent motif that varies depending on the species of the bacteria that you're looking at and the Cas9 that you're looking at. Varies depending on the bacterial species of the Cas9 gene. The most commonly used Cas9 nuclease derived from S. pyogenes. So the most common type of Cas9 that is used currently in vitro experiments is derived from S. pyogenes. And this was basically where it was initially discovered. And this basically recognizes a PAM sequence that has a sequence of NGG. 5' NGG. Sequence is always written from 5' to 3'. So if it is not mentioned, you have to take it that the first nucleotide is 5' and then it is moving towards the 3'. So this is N means any nucleotide, so it could be A, T, G, or C. The second and third nucleotide always have to be guanine. That is found immediately downstream of the target sequence in the genomic DNA on the non-target strand. So this is the non-target strand. This is the target strand of the protospacer. There's the non-target strand, and immediately downstream of this, you have the PAM sequence here, right? And this is, we are especially talking of S. pyogenes. In some cases, the PAM sequence has to be 3' of the protospacer. So that also we'll have to keep in mind, right? Cas9 nucleases with alternative PAMs have been characterized and successfully used for genome editing. It is important to note that PAM is not present in the small guide RNA, single guide RNA sequence, but a small guide RNA sequence, but needs to be immediately downstream of the target site in the genomic DNA. So whatever target DNA you have, the PAM has to be there in that target DNA, not in the CRISPR RNA, right? It has to be in the target DNA. Now, moving on, if you see here, how often are PAM sequences represented in the mammalian genome and on average? So using a frequency of GG of 5.21% in the reference human genome, there would be around these many 161, 284, 793 NGG PAM sites in the human genome on an average, or roughly 1 GG every 42 bases. So that allows you to roughly basically cover the entire DNA sequence, any particular gene that you want to target, unless it has a very extremely different average of GG as compared to the rest of the genome, you would still be able to target that particular gene, because a GG nucleotide will come every 42 bases, and N can be any nucleotide, so that is not a problem anyways, right? So therefore, roughly almost the entire human genome sequence can be altered using CRISPR-Cas, because naturally occurring GG positions on an average would come every 42 bases, right? Coming here, the difference between engineered CRISPR-Cas and the natural CRISPR-Cas, if you look here. So this is your natural CRISPR-Cas system. As shown earlier, you have a tracer RNA, you have a CRISPR RNA, and then you have a spacer region in the CRISPR RNA, which is complementary to the target DNA sequence. That DNA sequence is known as protospacer, and opposite to that, you have the PAM sequence here, right? What we do in artificial system is, instead of encoding this RNA from two different regions, we make a small single guide RNA. So this is called sgRNA, or a single guide RNA. This is not a dihybrid, it is a single RNA that we encode from a single plasmid. And what we also do is, since this region has to be common between all the RNAs, whatever gene you're trying to target, it is still the same. Only this region is to be variable. So we only allow this region to be encoded differently. The other regions are constant here. So in gRNA, it is known as guide RNA. Sometimes it is known as single guide RNA, small guide RNA. So in guide RNA, a short synthetic RNA composed of a scaffold sequence necessary for Cas9 binding and a user-defined 20 nucleotide spacer. So this region can be defined by the user. Whatever sequence you want to put in here can be put up, and therefore now whatever target you want to target on your target DNA, that can be targeted. So the guide RNA is a short synthetic RNA composed of a scaffold sequence. This is the scaffold sequence, which is common to all guide RNAs. Whichever gene you want to target, this will still remain the same. And then this is your spacer region, which is basically a user defined. You can define what sequence you want to have here, and that sequence will be put up here. So scaffold sequence necessary for Cas9 binding, and a user defined 20 nucleotide spacer or targeting sequence, which defines the genomic target to be modified. So this is basically the difference between the natural CRISPR-Cas and the engineered CRISPR-Cas that you have. In engineered CRISPR-Cas, you have a single RNA and a single protein. In natural system, you have two RNAs and one protein, Cas9 and CRISPR tracer RNA. And here in this case, you have Cas9 and single guide RNA, or guide RNA simply. You can also call it gRNA or guide RNA. The CRISPR tracer retroduplex is replaced by one chimeric RNA, single guide RNA, sgRNA, and the gRNA could be programmed to target specific sites. So this part of the guide RNA, this is customizable. You can put in any sequence that you want here, and then therefore now this becomes a targeted to that specific gene, right? So this is what is the difference between the two. Genome editing with CRISPR-Cas, engineered CRISPR-Cas consists of two components: a single guide RNA, sgRNA, CRISPR RNA, tracer RNA linked by a loop. So basically, you have a CRISPR RNA, tracer RNA which is linked by a loop. Basically, they represent CRISPR and tracer RNA, but generally it is one single RNA and a non-specific CRISPR-associated endonuclease, Cas9 or others. So this could be Cas9, a non-specific CRISPR-associated endonuclease, or it could be any other. It could also be Cas12. Nowadays, people are also using Cas12 very commonly. So the guide RNA is a short synthetic RNA composed of a scaffold sequence necessary for Cas9 binding and a user-defined 20 nucleotide spacer or targeting sequence, which defines the genomic target to be modified. Thus, one can change the genomic target of Cas9 by simply changing the targeting sequence present on the guide RNA. Now coming to CRISPR applications. So CRISPR can be used for gene inactivation. So you simply go to a particular, you can direct your CRISPR-Cas9 complex to a specific gene, to a specific position, and induce a double-strand break. When you induce a double-strand break, this break will be repaired by DNA repair enzymes, but the break repair by non-homologous end joining basically results in either insertion or removal of new sequences. So therefore, you have indels, and therefore, effectively, your gene will be broken up, right? And therefore, you will not be able to basically synthesize the protein. So you can induce a loss of function mutation using CRISPR here. This is most commonly done for studying gene function. Then, of course, if you see, you can have gene inactivation by large deletions mediated by non-homologous end joining. So here again, what you can do is you can induce large deletions. Basically, you target two CRISPR sequences, and they will cut at specific positions at in one chromosome on the same chromosome at slightly far distances from each other, and that would result in a large chunk being lost, and therefore you can have large deletions made by non-homologous end joining. And then, of course, what is more important here for genetic engineering and genome editing is that you want to basically correct an error, and therefore what you want to do is to replace a faulty copy of a gene by a correct copy. So in that case, what you follow is a homology directed repair, where you get a piece of, along with the CRISPR-Cas9, you also transfect into the cell a correct piece of DNA, which has homology to the region that you're directing for change, as we had discussed in earlier slides. And then what happens is when you do this, the double strand break is induced, and then this correct copy is replaced by the faulty copy here. And finally, you get a targeted gene insertion, wherein you can actually effectively target a piece of DNA and correct the errors, and then or insert a correct copy of the gene, insert the correct sequence, and replace the faulty sequence. So this is what we are interested in when you're doing CRISPR here, right? So class, you follow? Everybody there, class? All right. So Pratibha says yes. Then Sominal and everybody else says yes. Neha. So I'll take it that you're following, and we move on. So basically, CRISPR, the main application that we're interested in CRISPR is for correction of genetic inborn errors, and that can be done using homology mediated repair, which is this part here. And then, of course, if you want to study gene function in model organisms, you can induce mutations, single or small deletions, and that can be used. And if you want to see the effect of macro deletions or large deletions, that can also be done. But here, in that case, you'll have to use two copies of CRISPR directed far away from each other, removing a part of the chromosome or a part of or a large chunk of DNA, thus allowing you to study the effect of such large deletions. So this is our experimental paradigm, and of course, we want to use CRISPR for diagnostics. So if you see, this is one basic plasmid that is used for CRISPR-Cas experiments. If you see here, so you have your U6 promoter. This is the target region of 90 to 20 base pairs that can be modulated. So you can basically choose a sequence of your choice and put it here. This is the other scaffold part of the guide RNA, which will be common to whatever this sequence be. This will be common to all. And then, of course, you have in the same plasmid, you have a CMV promoter, and you have a T7 part of CMV sequence, coliform mosaic virus sequence, and then you have a T7 promoter, and then you have Cas9, which is also expressing along with it in fusion a green fluorescent protein, so that you can follow where your Cas9 is moving around if you want to do an experimentation and want to see how Cas9 is moving around and whether it is its location and its expression levels. So if you want to do that, because in biotechnology, it is very important that you keep checking at every step that whatever you're doing is actually happening or not, right? So for that, you put up these fluorescent molecules. So basically, you can tag a protein with GFP. So it will give you an idea whether your protein is getting expressed at all or not. You can also follow the protein as it moves through one compartment to another compartment. You can see its localization. And if you basically tag two different proteins with different fluorescent markers, you can also see if they're interacting or whether they're expressed at the same time or what are their expression levels with respect to each other and so on and so forth. So here in this case, this is your CRISPR-Cas single plasmid format where you express your target region, the guide RNA and Cas9 in a single plasmid. So this portion remains exactly the same. The scaffold portion of guide RNA is also the same. And then this 19 to 20 base pair region is where you have to basically specify which target you want to put up here. So that complementary sequence comes up here. And then, of course, once you have your single guide RNA here, so this is your single guide RNA, you have regions of complementarity, you have the unpaired region here, and this is your target specific guide RNA sequence, CRISPR RNA sequence, as to say. This then combines with Cas9 and scans your DNA for complementarity, and wherever it finds complementarity along with the PAM sequence, there's going to cut here, and then of course you can have cellular response in terms of repair, and when the repair happens by non-homologous end joining, you will have loss of a bit of a sequence resulting in loss of gene function. Now coming to a brief history of CRISPR. So this is, if you see, they were first described way back in 1987, and until the next 13-14 years, they were just part of history, and there was not much that was happening here, right? So CRISPRs were described way back in 1987. It's like your papers by Mendel. So they were described much time ago, and they were lost to history, and then only in 1901 around that they were rediscovered by the three different individuals, and then of course they're taken forward. So likewise, CRISPR, you may have a basic discovery whose applications may not be understood for a very long time, and then suddenly a sudden turn of events can result in, you know, identification of the application of your basic research. So basic research is absolutely essential for applied research. Then of course 2005 and 2006 is CRISPR contains viral sequences, Cas genes, identified hypothesis. So basically around 2005, 2006 was the first time that people understood what is the significance of the CRISPR array. People knew that there is a sequence of, you know, repeats, and it is broken by a certain non-repeat sequence in between, but no one really understood what this was basically representing. And then suddenly around 2005-06, people realized that these are basically representing the viral infections into the bacteria, and that this is the adaptive immune system. And since then, CRISPR has been growing very strong, and 2020 award has been given to CRISPR technology. The Nobel Award has been given to CRISPR technology. And then, of course, if you see, why is CRISPR-Cas so successful, and why is it being used for not just genome editing, but beyond, for introducing methylation changes, for introducing other kind of gene upregulation, gene downregulation, as also for for diagnostics. It is because it is like a Swiss knife. It is simple, it is elegant, it is customizable, and it is modular. So that is what allows it to be used everywhere and every time for any specific purpose that you want to.